So my name is Jack. I'm an educator here at the Regeneron DNA Learning Center, and thank you for coming to this series, our Meet a Scientist seminar series. We're really excited to have our speaker today, Terrence Turner, who's a senior scientist here at Regeneron Pharmaceuticals. So again, we thank him for taking his time to speak with us today. Before we jump into his talk, I would just want to share some information on some of the programming that we have at the Learning Center coming up this summer. So for this summer, our summer camp registration is open for 2023. We have summer camps being offered at all three of our locations. So out on Long Island, at our Brooklyn location, as well as at my location currently at the Regeneron DNA Learning Center up in Sleepy Hollow, New York. If you're still looking for virtual options, we still have some virtual options for you as well. But if you come to our Science Summer Camp page, you can take a look at the schedule for all three locations. You can see which dates work for you best. Hopefully we can find something that works for you. And then I would also just recommend checking out our reference guide just to make sure that you're signing up for a camp that is age appropriate for yourself or your child. And then I also want to just bring your attention to a couple other things on our public page. We do have Saturday DNA sessions being offered at our three locations. So you can check out, see which ones are being offered. We have ones this Saturday out in Long Island, as well as here at Sleepy Hollow. And we also have some new school break bio sessions. So these are sessions being offered during spring breaks for most students. So if you're not going away and you're looking for something to do during the week, we do have some of our spring break bio sessions available. Those are in April. So when most students are hopefully on spring break. So if you want to check those out, we do still have spots open for those. So those are most of the announcements that I wanted to take care of before we get started. And again, we have Terrence Turner joining us. He's going to tell us about DNA sequencing in the modern age. So Terrence, take it away.
Thank you very much, Jack. Let me share my screen. Okay, hello again, everyone. And thank you very much for having me here today. Just going to tell you guys a little bit about sort of my career path, as well as what I do here as a scientist at Regeneron. My title of the talk is DNA Sequencing in the Modern Era. My early life, I grew up in Flatbush, Brooklyn, and I, learned hard work from great hardworking parents. And I grew up with some good friends that were very motivational and driven. And I just simply avoided a lot of troublemakers growing up in Brooklyn in the 90s, where there was certainly a lot of that around. And then when I got to undergrad, I went to Marist College, which is a small private school up in Poughkeepsie, New York. And I started out there as an athletic training major, ended up switching into biomedical sciences. And then I really enjoyed chemistry and took all of the chemistry courses offered in the school. And they actually gave me a chemistry major in my junior year. So I ended up becoming a biomedical sciences and chemistry double major in undergrad. I was also a football scholar athlete. I was two-time captain of the football team and academic All-American, and had some all-league honors in college playing football as well. Also in undergrad, I did a lot of undergrad research. I always had an interest in research, and it's why I picked the career that I'm doing now. So in undergrad, I did a lot of research in my college at Marist College. I also did a summer undergrad research opportunity at Albert Einstein College of Medicine in the Bronx. And I was able to get co-authored on 2 papers while in undergrad. The first one was studying epigenetics back when it was still pretty early in the study of epigenetics, where sequencing wasn't as powerful as it is now. And the other project was studying and identifying new species of silverfish insects from all over the Caribbean islands. And so got interested in sequencing very, very early on too, with these different experiments. And then my experience in the biotech industry started with my entry-level experience, where I was a temp research associate at Regeneron. This company was the first company I ever worked at. And at that position, I was working in a QC group. And so a lot of times you have a lot of repetitive processes that have to happen inside of different research labs and research departments. And that position was mainly to, QC and quality check all of the different constructs that were being made before they were delivered to the next labs. And from there, actually, my contract ended at Regeneron, and I went over to Pfizer Pharmaceuticals, where I was also in a sort of entry-level position too. And at that position, I was more in vaccines research. I was running these automated liquid handlers. So these are automated robots that were able to do serial dilutions for ELISA assays. And so I actually worked on Pfizer's Prevnar 13 vaccination at the time before I came back to Regeneron, where I am now in my current role, where we produce modified animal models. So we make modified alleles inside of the DNA of mice for the purposes of making them model different disease systems in humans, so that we can identify and evaluate therapeutics inside of these modified mouse models. And so to do that, we use these small molecular constructs of DNA, and we actually micro-inject them into the mouse embryonic stem cells. And we do it at a very particular stage where we can almost guarantee that all of the cells that will be generated from those ES cells will all have the modification that we intended to put inside of these mice. And so what's really nice about my position right now at Regeneron is that I get to, design a lot of my own experiments. I can investigate things that interest me and things that are in the interest of the group and the department. And now I'm doing a lot more work with long read Oxford Nanopore sequencing. I'm going to talk a little bit more about what that is, if anybody doesn't know. But it's a new, relatively new and very powerful piece of technology that allows for very long sequencing reads that allow you to make these alignments inside of the genome and find different positions where there's modifications in the genome. And so right now, this year I've been named the lead in these efforts in my department. And so I'm rolling out a lot of different processes and doing a lot of bioinformatics as well as wet lab processes to get these reads and get the answers of what we're trying to modify in the genome. And so what I was talking about before when we were making those recombinant DNA vectors, so we start out with these cloning expression vectors on the top left, and we will digest them with restriction sites, or restriction endonucleases, or we would digest them with Cas9, CRISPR-Cas9, in very specific locations, and we would introduce different DNA fragments into these vectors and then have them ligate together. And then this allows us to make these recombinant plasmids with a new piece of DNA. And that DNA can contain a whole gene or just part of a gene that we want to put inside of the mouse embryonic stem cells. So it's a very powerful technique that we use, and it works very well for developing these modified alleles. Okay, so now this is a quick overview of the Oxford Nanopore sequencing technology. So Oxford Nanopore is a company that started in Oxford, England, about maybe 10 years ago. It's relatively new in the sequencing world, but it's very powerful. And that is because of one detail that it has, in that it can produce very long reads. So what it does basically is it takes the double-strand DNA molecule that you see up top, and it will feed the molecule into this nanopore. And so this is an engineered protein that has been engineered so that the barrel of the protein is only the width for one single strand DNA molecule to pass through at a time. And then there's a current, there's a voltage difference between the top and the bottom of the nanopore that is embedded in a membrane. And so as the molecule passes through the pore, it's actually measuring the voltage that's passing as ions are passing through. And the molecule of DNA will actually disrupt the voltage as it's passing through. And that disruption is what you're measuring in the current intensity. And then based on the nucleotides that pass through, they each give out a different profile of disruption of that current. And these profiles are small electrical signals that can be called with very complex computational algorithms that are able to tell which nucleotide passed through at a given time. And so measuring the nucleotides that pass through over time is the act of sequencing. And so when I found out about this technology, I was very impressed, because unlike traditional methods require amplification for sequencing, where you'd amplify with like Sanger or NGS. As you're amplifying, you're adding a fluorescent molecule that will give off a signal. This is not amplification at all. You're actually reading the DNA molecule itself, and you're getting these signals from the DNA molecule. And this allows you to not be limited to the read length associated with amplification, because amplification can't really amplify forever. The polymerase will fall off. And so because it's going through the pore, you can actually read the entire molecule. And we've read up to almost 1 million bases at a time on one read, which is very long. And so as I mentioned, this gives like really long read data sets, and we can get millions of bases in a whole library. The very cool thing too is we can also add these sequencing adapters. So we can start the sequencing at very precise positions inside of the genome and sequence in certain locations that we're really interested in. And so that's what the image is showing in panel A there, is that you're adding sequencing adapters at special spots on the genome, and then you're originating your sequencing there, and then it's passing through the flow cell. And in panel C there, you're actually, that's the device. And so it's really cool. In their website, they have a lot of information about people using these devices because they're portable, and you can just plug them into a computer in the USB People have used them in space. They've used them in the farms and out in the fields in different areas, sequencing plant genomes and finding contaminants in crops and things like that. So it's very powerful in that it's also portable. Not only is the technology really good for the sequencing calls and the accuracy and the read length, it being portable allows it to be used for a bunch of other things too, which is really neat. And so this is just so this is real data that I've generated. And this is just an appreciation of the read lengths themselves. And so if anybody's ever familiar with doing like PCR or some other sequencing techniques, you'll see that getting reads up to 166 kilobases is really large, versus PCRs are typically under 10 kb, and Sanger reads are under 1 kb usually. This is just to give you an appreciation of just how long these reads are. And these are not even the longest reads we've gotten. Oops. And so what we're using these nanopore sequencing to do is we can actually identify copy number variants and structural variants. And so these are just some cartoons demonstrating the different possibilities that DNA sequences can kind of rearrange inside of the genome. And so if you think of each one of these like little blocks as, tens of thousands of base pairs, you can actually get whole copies of big chunks of DNA that are duplicated or cut out or relocated into different positions and all kind of different combinations. And because these are long reads, you can capture all of these different situations in a single read. And with some of the algorithms that we're designing and we're using in our group, we can actually recognize some of these things, which is pretty exciting. And so this is some live data from what we've done with ONT reads. And so there's this very particular region inside of the genome called C9orf, and it is characterized by these G4C2 repeats. And it actually leads to really bad degeneration of your neurons when you have a lot of copies of these G4C2 repeats. And so we're able to use long reads to go all the way across these repeats and actually count how many repeats there are in some of the models that we're making. And so this is just another example of where we can see exactly the length of the repeats and see if they're not all G4C2 or if they have retractions or expansions and things like that. And so what Regeneron basically does is we make monoclonal antibodies. As I mentioned, we're making these mouse models to be able to test these monoclonal antibodies inside of our mice. But when we're making our therapeutics, we generate these antibodies that can be used for all different types of purposes. We have different targets, anywhere from cancer to different muscle disorders and a lot of other things. And this is actually the Regen COVID treatment that happened in 2020. I actually worked on during the lockdown when we were doing the ACE2 knockouts here at Regeneron. So that's what we are doing. And so I just want to mention that you guys are in the middle of what I believe is a scientific revolution. I think it's the convergence of a lot of different, very powerful scientific breakthroughs are happening at this current time. DNA sequencing with methods like this, and there's a bunch of other different techniques that are coming out. Even the traditional methods are actually getting a lot better with DNA sequencing. So we're getting a lot more DNA data. Regeneron actually has the Regeneron Genetics Center, which is sequencing exomes of patients from all over the world and correlating that data to clinical data and finding correlations between the genome and patient information, phenotypes, like literally identifying phenotypes, which is very powerful. And then AI, I'm sure if everybody's heard recently with Chat GP3 and GP4 and all these different AI models that are coming out. AI is going through a rapid change right now. Energy storage, batteries are getting cheaper and bigger and faster, and energy storage and generation is getting a lot better. Computational abilities. Every couple of weeks, it feels like there's a new chip, a processing chip that comes out, and it can do a lot more computational power as the ones before. And then AR and VR are going to be huge in your careers as you get older and you get further into science. And so what I think is unique here and probably not talked about very much is the fact that all of these things are happening at the same time, and they're actually feeding off of each other and they're converging. And so because the computational abilities are better, DNA sequencing is actually getting better. And because DNA sequencing and the computational abilities are getting better, we're using AI in order to be able to identify different targets and find out different things about the genome. And then, so all of these things, you can find different areas where these relationships are being built between these different technologies. And this is just the 5 that I've identified are the biggest that I'm really looking at into the future, but I'm sure that there's many more. And some words of encouragement I give to you all. Number 1, invest in yourself. I can't overstate how important that is. I think that it's important for you all to find something that you're interested in. For me, it's the sequencing and computational stuff. I'm kind of devoting my entire career to it because I like it. And sometimes it doesn't even feel like work. It feels like a hobby where I'm sitting down and I'm doing computational work, and I'm in the lab, and I'm, extracting samples, and I'm figuring out a way to do different things. And so I've invested in myself in building on my computational skills. I don't have a computer science background, but I was able to go in and learn on my own how to code and do things like that. Build your team. When I say build your team, surround yourself with people that help you, motivate you, push you, and things like that. That's family and friends. And when you get into college and further on in your careers, you'll see that there's a lot of people that want to help you, and you can surround yourself with people like that will push you and motivate you. Keep an open mind. Keep an open mind of the different career paths that might come about and the different opportunities that might show up in terms of areas that you may not think you would be interested in. But if you just go to a seminar here or there, or you listen to a lecture, you never know, you might be really interested in it. And enjoy yourself, enjoy the ride, enjoy learning, enjoy working hard, and please don't give up. There's gonna be really tough times where things aren't working, especially in science. Almost 50 or 60% of the time, things don't work, but you got to just keep pushing and keep going. And so with that, I thank you all again for hearing out the talk, and I we'll open it out for questions.